How quantum is life? Imagine taking the most advanced quantum computer humanity can build and placing it inside a warm, wet rainforest. No cryogenic chamber, no vacuum shield, no vibration control. Within moments, its quantum states would be overwhelmed. Heat would scramble them, moisture would disturb them. Every tiny collision would push the machine toward failure. But now look beside it. A leaf is absorbing sunlight, a bird is crossing the sky, an enzyme is changing the speed of life. And in each of these systems, biology may be using processes that are sensitive to quantum behavior. That is the paradox. Human quantum machines need extreme isolation. Life works in the storm. So the question is not whether life is magical, the question is sharper. What does life know about quantum survival that our machines still do not? And could the answer reach deeper than biology itself? In the laboratory, quantum behavior is treated like a rare and delicate performance. A particle can exist in more than one possible state. A wave can explore more than one path. Two quantum states can keep a precise relationship in phase, but only if the system is protected. Too much heat, too much vibration, too many collisions, and the fragile relationship disappears. This is decoherence. It is the process by which quantum possibility loses its sharpness and the world begins to look classical again. That is why quantum engineers build cold, isolated, carefully shielded machines. But biology is the opposite of that machine. A cell is not cold. It is not quiet. It is not isolated. It is crowded, vibrating, chemically active, and constantly exchanging energy with its environment. By the simplest intuition, a living cell should be a quantum graveyard. And yet, in several biological systems, quantum-sensitive behavior keeps appearing, not as science fiction, as a real experimental question. The first fingerprint appears in photosynthesis. When light strikes a photosynthetic complex, energy does not simply hop like a ball from molecule to molecule. In some models and experiments, it behaves more like a wave moving through a network. The energy seems to explore possible routes before reaching the reaction center with remarkable efficiency. The second fingerprint appears in animal navigation. A leading hypothesis suggests that some migratory birds use proteins in the eye called cryptochromes. Inside these proteins, light can create pairs of radicals. Their electron spins may respond to the weak magnetic field of Earth. If this mechanism is correct, the bird's compass is not a tiny metal needle. It is chemistry balanced on quantum spin. The third fingerprint appears in enzymes. Some reactions may be accelerated because particles do not climb over an energy barrier. They tunnel through it. Each example is different. A leaf is not a bird is not an enzyme is not a quantum computer. But the same question keeps returning. How can warm biological systems preserve enough quantum structure to make any of this useful? Before we go deeper, we have to be fair to mainstream science. Biology does not need to violate physics. In some systems, the environment itself may help. Thermal noise can sometimes shake energy loose from a bad path. A protein can shape the local chemical environment. A molecule can protect certain states just long enough to matter. This idea is often called environment-assisted quantum transport. It is a serious idea. It says noise is not always the enemy. Sometimes, if the structure is right, noise becomes part of the route. But the deeper question remains. Why are some biological structures so good at this? Why do certain molecular shapes seem to sit exactly at the boundary where quantum fragility becomes biological function? This is where the PTQ perspective enters, not as a replacement for biology and not as a proven theory of life, but as a geometric question underneath the biology. PTQ begins with a careful distinction. It does not ask us to imagine a fifth direction in space. It does not ask us to walk into a hidden dimension. Instead, it asks whether the physical world we observe is only a projected sector of a richer underlying structure. In ordinary geometry, a point in spacetime tells us where and when something happens. A flash of light, a molecule absorbing a photon, a particle changing state. But in a PT-symmetric quaternonic picture, the deeper kinematical structure may be richer than what is directly observable. The key is projection. Not every internal degree of freedom becomes physical. Not every mathematical structure becomes measurable. Physical meaning appears only after an admissible projection selects an observable sector. That is the important boundary. PTQ does not say that hidden structure is automatically real in the laboratory. It says that if a residual structure survives projection, then it may leave controlled, observable traces. In this picture, spacetime is not only a smooth stage, it may have internal grain, but that grain is not directly seen. Only its projected residue can matter. Now return to biology. If a quantum state evolves inside a warm molecule, its phase can drift. Thermal noise pushes it off balance. The environment tries to scramble the pattern. In a PTQ-compatible extension, any correction would not come from a new substance, not a glowing fluid, not a mystical energy, not a special life force. It would come from the way observable dynamics are projected from a richer underlying geometry. If a residual geometric structure survives that projection, it may slightly change the effective transport of phase information, not as a miracle, not as a biological privilege, but as a constraint on what counts as stable, observable evolution. At this point, a skeptical question should appear. If this geometric residue exists everywhere, why does a quantum computer still need extreme cold? Why can a leaf work in sunlight while a machine worth billions needs a refrigerator colder than deep space? The answer is not that life has access to a secret field that machines cannot use. The safer answer is this. Engineered qubits and biological molecules may occupy very different dynamical regimes. Human quantum engineering usually tries violent isolation. Remove heat, remove vibration, remove noise. Build walls around the quantum state. Life cannot afford that. A cell cannot become a vacuum chamber. A bird cannot cool its eye to near absolute zero. So evolution uses a different strategy. It does not build thicker walls. It searches for better shapes. A good analogy is sound. If you need to send a whisper across a noisy valley, one solution is to build walls everywhere, block every echo, suppress every disturbance, control the entire environment. That is the engineering solution. But there is another way. Find the natural echo of the valley, stand at the right point, use the shape of the landscape. Then the whisper travels farther than it should. This is the evolutionary solution. In the PTQ interpretation, biological molecules may be viewed in a similar way. A photosynthetic complex is not just a chemical assembly. It may behave like a resonator-like structure, a shape that helps energy move through a noisy environment. A cryptochrome protein is not just a pigment. It may be a molecular stage where light, chemistry, spin, and magnetic direction meet. The point is not that life discovered a magical force. The point is subtler. Evolution may have selected shapes that sit unusually close to the conditions where quantum information survives. If spacetime has internal texture, then the best biological structures may be the ones whose ordinary quantum dynamics are unusually sensitive to the projected residue of that texture. But a theory like this cannot survive on beauty. It needs places where nature can say no. The first test is cosmic. If the same projection-induced residual structure appears at large scales, it may leave relations between galaxy dynamics and the expansion scale of the universe. This is not proof that dark matter is false. It is not a shortcut around cosmology. It is a dangerous prediction. Two mysteries that appear unrelated may share one geometric scale. That can be checked. The second test is biological. Take the molecule most associated with the bird compass. Place it in a controlled laboratory setting. Rotate its orientation. If the effect is purely local chemistry, the coherence lifetime should not reveal a clean geometric direction. But if a projected residual geometry contributes, the lifetime may change periodically with angle. Angle the molecular resonator correctly and the signal strengthens. Turn it away and the signal weakens. This is the kind of test that matters, not because it sounds poetic, because it could fail. Around these two anchors, the framework points to other possible fingerprints. A subtle drift in cosmic expansion history, a phase shift in neutrino oscillations, a tiny high-frequency dispersion in gravitational waves, a resonance-like peak when biological molecules are coupled to quantum sensors. The exact details are not established. The point is the logic. Different scales, different instruments, one question. Does nature repeat the same projected geometric signature? If each domain requires a separate explanation, the idea weakens. If the biological effect needs one structure, the galaxy effect needs another, and the quantum sector needs a third, then the unified framework fails. That is not a small problem. That is the test. PTQ stands or falls on cross-scale consistency. So what would this mean if it were ever supported? Not that life is magic, not that consciousness controls reality, not that biology escapes physics. It would mean something quieter and perhaps deeper. Life may be one of the places where the structure of physics becomes unusually visible. A quantum computer survives by being isolated from the world. Life survives by being open to it. That is the strange inversion. Cells do not silence the storm, they build shapes inside it. Leaves catch light, birds cross oceans, enzymes reshape chemistry, and perhaps, at the deepest level, these are not separate miracles. They are experiments performed by evolution on the geometry of matter. The PTQ hypothesis may be wrong. The projected residual structure may never be found. The biological signature may vanish under better experiments. The cosmic links may fail, but the question remains valuable because it forces us to look at life differently, not as an exception to the universe, but as a detector, a soft, warm, fragile detector built by evolution, testing what kinds of quantum order can survive inside reality. So how quantum is life? Perhaps not quantum in the way science fiction imagines, not a glowing force, not a cosmic mind, not a hidden life energy. The deeper possibility is more disciplined. Life is made from matter. Matter exists inside space-time, and observable physics may be a projected sector of something richer than our simplest pictures allow. If that is true, then life is not outside physics. It may be one of the most delicate instruments physics has ever built. The question is not whether this vision is beautiful. The question is whether nature leaves the same signature twice, in the eye of a bird, in the spin of a particle, in the light of a distant galaxy, in the trembling geometry of space-time itself. Until then, the claim remains open. But the question is worth asking. What if life is not merely happening in space-time? What if life has been testing how much quantum order space-time allows?